Laboratory 6 is a small-scale representation of the final steps in the overall process of recombinant therapeutic protein production. The first step in Lab 6 is creating a liquid bacterial culture. This step is primarily done by the teacher. There are a few different methods that ABE sites use for this step. Some sites suggest that teachers pluck a pink colony from a student LB ampicillin arabinose plate and inoculate a flask. And some sites supply bacterial cultures already expressing RFP. Please check with your site technician or site coordinator to learn how your site supports Lab 6. Regardless of how you obtain your culture, the student experience starts with the lysing of these cells to release the RFP protein from the cells into the supernatant. One teaching tip is to remind students to think about where the protein is at all times. When students first obtain their sample of culture in the EC tube and are asked to centrifuge the sample down, a cell pellet forms at the bottom of the tube. Since the protein is still enclosed in the cell, the condensed pellet of cells is pink. Once students break open their bacterial cells with lysis buffer, they are asked to centrifuge once again. This time, the RFP will be in the supernatant, or at the top of the tube in the liquid, and the cell debris, now white, will be pelleted at the bottom of the tube. Similar to Lab 5, safety and sterile technique should be stressed to students. You should be given a spray bottle of disinfectant, whether bleach, ethanol, or another solution, depending upon your ABE site. It is good practice to clean the lab benches or desks, both before and after the lab experience. There is also waste that must be discarded appropriately. Please contact your ABE site technician or coordinator to learn your site's protocol for waste. Keep in mind that you must also abide by any school or district waste disposal regulations.